sir?
Yes, yes.
Yes.
Am I
So
audible?
now, yes, sir. So now I request Twinkle to kindly introduce our eminent speaker. Over to you, Twinkle.
Good evening, everyone. Thank you, ma'am, for the opportunity to introduce our esteemed speaker, Dr. P. Gopinath. He is a professor in the Department of Biosciences and Bioengineering in Indian Institute of Technology, Roorkee. He earned his PhD in biotechnology from the Indian Institute of Technology, Guwahati. He did his postdoctoral research at University of Rochester Medical Center, New York, USA. His research group in the Nanobiotechnology Laboratory at IIT Roorkee is working on developing various nanomaterials for biomedical applications. At present, he has more than 150 research publications in the area of nanobiotechnology in high impact factor journals. He has filed 30 patents in which 17 are granted and done one technology transfer. He has also published 8 books and 15 book chapters. His research work has been well cited with total citation of more than 9784 till date. Further, he has received several prestigious awards including Tata Innovation Fellowship by Department of Biotechnology, Government of India BIDAC GYTI in 2019 from Shri M. Venkaiah Naidu, Honorable Vice President of India. He is a Fellow of Royal Society of Chemistry, United Kingdom, Fellow of Royal Society of Biology, United Kingdom, and Fellow of Academy of Sciences, Chennai. He is the co-founder and director of startup company Super Good Nano Private Limited. With that, I now invite Professor Gopinath to kindly take over this session. Thank you, sir.
Yeah, thank you. Thank you very much for the nice introduction. Uh, Let me share the slide and hello.
Yes, sir. You are audible, sir.
Yeah, somehow it's stuck actually. Uh, so my slides are visible in a full screen.
Yes, sir.
Yes, now it is fine.
Okay. Oh, just 1 second. It is. Yeah. So very good evening. So my voice is clear, right? Audible. Everything is fine.
Yes, sir. Yes, sir.
Okay. Okay. So the title of my today's talk is on biomimicry to translational nanobioengineering concept and applications. I thank Dr. Shilpa for providing this wonderful opportunity and the organizing committee members. And today I'll be talking about slides. Yeah. So today I'll be talking about what is nanoparticle and why small is good and what is the advantage of the surface area to volume ratio and also how to make these nanostructures, and I'll be briefly talking about what is nanobiomimicry and what kind of technology we developed in our lab, so that also I'll briefly go through. So let us see what is nanoparticle. So most of you know the nanoparticle means any material having at least any one of its dimensions should be in the range of 1 to 100 nanometer. For example, if you take any material, it will have a X, Y and Z. So out of this, at least any one of its dimensions should be in the range of 1 to 100 nanometer. Then it is called as nanomaterials. So we have 0D, 1D and 2D nanomaterials. And the word nano means, it's a Greek word that means small. And the term nanotechnology was coined by the scientist, Japan scientist Norio Taniguchi. And most of you know the nanometer means it's a 1 billionth of the meter, that is 10 power minus 9 meter. To make it more simple, so we can compare our Indian population. For example, our Indian population few years back, it is like 100 crores, that is 10 power 9. So when you compare yourself to the whole Indian population, you and me are nano size. So that much small is the nano. So we can also see in another simple example, that is like it is equal to 1 rupee in 100 crore 1 rupees. So that much small is the nano size. So why small is good? We already have different sizes. So what is the advantage of this small? So if we see that, so this is a 7 shells and it is made up of total of 1400 atoms, and if you see the surface atom is only 35. Whereas if you break the same 7 shell into one shell, so you can see that the total number of atoms is only 13, but the surface atom is very high. So that's why whenever the size goes down, the surface area to volume ratio will be higher, and the objects can be faster, and it can be lighter, and it can be more energy efficient. So that is the advantage of these nano materials. And again, what is surface area to volume ratio? Whenever we talk about nanoparticle, we always say that the size goes down, the surface area to volume ratio will be higher. So we can see this example. We can take a 2 mm box, and the same box can be cut into 1 mm box. Then we can apply the surface area formula. Then you can see that the surface area for this is higher when compared to this. So assume this is like your bulk material, and this is like your nano material. So the same bulk material, when you make it into nano, the surface area is increased, but the volume is same. So that's why the surface area to volume ratio will be higher when the size goes down. And additionally, the advantage is when you break this bulk into nano, what happens is all the hidden surfaces are also exposed. So this is your bulk, and this is like your nanoparticles. So when you make it into nano, so all the hidden surfaces are exposed. So this results in a better catalytic activity. So for example, if you are using this aspirin tablet or Eno tablet, you just put in the water. It will take 50 seconds to dissolve. And the same Eno tablet or aspirin tablet, you break it into powder and put in the water. Within 10 seconds, it dissolves. So when the size goes down, the surface area to volume ratio will be increased. Then also all the hidden surfaces are exposed. So it can have a better catalytic activity. So that is the advantage. And again, the material properties also vary with respect to the size of material. For example, the bulk gold, which you use to make ornaments or jewels, so that when it goes to the nanoscale, so it will show some remarkable properties. For example, from the yellow color, it becomes like purple and become ruby red color, and when it's reached to the atomic scale, it becomes colorless. So with respect to the size, with respect to the shape, it gives the beautiful colors. So the reason for this beautiful colors is SPR, that is the surface plasmon resonance. And again, similar to the biotechnology, so it is not a new field. Similar to that, nanotechnology also not a new field. If you see that the olden days, like in the churches, so they made a lot of beautiful paintings. So those days they don't have any branded color paints, but they had a lot of gold and silver. So using the gold and silver, they made these many beautiful paintings, and with respect to the transmitted light and reflected light, it will show the different kind of color. And these gold nanoparticles are highly stable, and these nanoparticles are stable, and it was destroyed during the World War II. So it is highly stable nanoparticles they made those days. And again, if you see this Lycurgus cup, so those days they don't have any LED light or any graphics, but they made this kind of Lycurgus cup with the help of gold and silver coating on the dichroic mirror and the glass. And with respect to the light, okay, so it will be appearing red color in the transmitted light, and it will be appearing green in this scatter light. So by this way, this can be also very useful, and like using the gold and silver, they made this kind of Lycurgus cups. And additionally, the advantage is the silver also has an antibacterial property. So they use it as a kind of antibacterial coating, as well as for making this the cup as like a special cup for the king. So that is the advantage of this gold or silver nano. And it is also not new to the Indian traditional medicines like Ayurvedic medicines and basmas. They can see that. So the basmas also, they use the gold and silver collide. So how to make this nanostructure? There are two approaches. One is top to down approach. The other one is bottom-up approach. So the top-down approach is from a very very big size you are going to make the nano particle. So from the bulk you are going to make these nanomaterial. From the rock you are going to make the statue. So that is called top-down approach, from a very big size to smaller size, top to down. And another approach is bottom-up approach. So in the bottom-up approach we are going to use a very very small size and you are going to make nano size. So most of the biological materials comes under the bottom-up approach category. So for example, if you take the DNA, so DNA is made up of nucleotides, ATGC, and if you see the size of each nucleotides, which will be in the range of angstrom. So this nucleotides combines and form the nano size DNA. So that is called bottom-up approach. So from a very very small size, you are going to reach the nanometer size. So you are going to use the brick and construct the building. It is similar to that, like you are going to use these nucleotides as a building block. For example, the proteins are made up of amino acids. So amino acids are the building blocks. So assume this brick as amino acid. So the size of each amino acid in the range of angstrom. So it combines and forms the nanoscale protein. So this is called bottom-up approach category. So from a very very small size you are reaching to the nano size. So what is the difference between nanoscience and nanotechnology? So if you are making a silver nanoparticle or if you are making a fluorescent nanoparticle, and if you publish a paper or if you publish a thesis, that is a nanoscience, and the same quantum dots or the fluorescent nanoparticle and you are using and making a product, then it is a nanotechnology. You can use the nanosilver based bandage, or you can use the nanosilver coating, or using the quantum dots for making the quantum LED TV, that is the nanotechnology. And what is the difference between nanobiotechnology and bio-nanotechnology? So most of the time these two words are interchanged or used. Nothing wrong in that, but the clear definition is nanobiotechnology means applications of nanomaterials for biological applications. For example, so if you are using a nanomaterials for cancer diagnosis or cancer therapy, that means you are using nanomaterials for biological application. So that's why it is called as nanobiotechnology, nanomaterials in biological application. And bio-nanotechnology is understanding biological nanostructure. For example, I told you this DNA and protein, so these are also in the range of nanometer. So can we use this DNA and protein and make different nanostructures for various applications? So that is bio-nanotechnology. So understanding the biological nanostructures. So we got a broad idea about what is nanomaterials. Now I'll explain about what is nanobiomimicry. So what is mimicry? So most of you know mimicry means it's copying or imitating. So we might have also imitated or copied during our school days, college days, our teachers. So that is called mimicry. We are imitating or copying someone. And what is biomimicry? So biomimicry means you are copying some idea from the nature. So why we have to copy the idea from the nature? So if you copy the idea from the nature, the success rate is always higher, because the nature has done several millions of year experiments and made the final product. So for example, you and me are successful product of nature. All the living organisms, if you see that, so the nature has made it. So nature has done millions of years of experiments and optimized the parameter. So for example, whenever we need help, we take our seniors' help in the lab, because they have optimized the experiment several times. So they might have done 3 times or 5 times, but nature has done millions of times and made the final product. So that's why when we copy the idea from the nature and make the product, the success rate is always higher. So the simple example for biomimicry is, you can see that we can take the idea from the bird and we can make the aeroplane. So this is a simple example for understanding the biomimicry concept. So similarly, there are plenty of examples, like for example, the scientists got the idea of the train from the snake and millipedes. So there are several other products also. Even the Japan bullet train, they got the idea from the kingfisher. Okay, so there are plenty of examples. These are examples for the biomimicry. So let us see what is nanobiomimicry. So nanobiomimicry means we are going to copy the idea from the nature, but at the nanoscale level. So the first example is like we can take the example of gecko lizard. When you see the gecko lizard, it claims on all the smooth surfaces and it won't fall down, and it stick firmly to the surface. And when you see that, what is the reason? So when you see the leg under this electron microscope, it has a nanostructures, and this nanostructures is providing the grip to this gecko lizard, and it is able to climb even the smooth surface or slippery surface. And if you see the interaction, it is mainly due to the van der Waals forces. So from that, some scientists made different products. So the first product is they made a biocompatible patches, and which can be useful kind of wet adhesive. So for example, whenever our cycle tube get punctured, so we go to the shop and the puncture person, he completely dry that tube before he put the glue and stick the or close the puncture gap. So but if there is any blood vessels got damaged, so we cannot drain out the blood or we cannot dry the surface. So we need a wet adhesive which can close the wound or seal the gap. So for that they made a wet adhesive, and again it's similar to the velcro. So you might have used the velcro shoes or velcro laptop bag. So this biopolymer is a kind of velcro kind of things, and you can apply this wet adhesive glue, and it is made up of two biomolecules, glycerin, and it can be activated by UV light. So when you apply this glue and apply the UV light, it will cross link like a velcro and seal the gap, and we no need to dry the wound. It can work in the wet environment, so that is the advantage. So they got the idea from this gecko lizard, so that's why they made a company also, Gecko Biomedical. And another company, they got the idea from that and they made the gecko shoes. It will be useful for the people, those who are climbing or mountaineering and trekking. And not only for them, it will be also useful for the old age people. So those who have the walking disability or the problem. So nowadays, if you see that most of the homes are made with the tiles and this thing, so it has already having a slippery nature. So this kind of shoes will have a more grip and it will provide the balance for those people. The another example is like a chameleon. Like we can make a color changing material, so it can be like act like a camouflage, and we can copy the idea from this chameleon and make such kind of dress materials which can be useful for the person who is in the defense in the army. So that can change the color according to the nature of the environment. So we can copy the idea from this chameleon and we can make these products. So they made the product by using this silicon layer of 120 nanometers thick, and with respect to the light, the color will be changed. So another one of the important aspect is like, for example, recently we faced the COVID pandemic. So the next pandemic the scientists are expecting is like AMR. So there is a antimicrobial resistance. So for example, like previously whenever we get any bacterial infection, if we take antibiotics, it gets cured. But nowadays we have to take multiple antibiotic doses and combination of several antibiotics. So slowly the bacteria is getting resistant to all the antibiotics. So what is the alternate way? Whenever you make any new drug, so after a few years, the bacteria is trying to develop some alternate method to escape from these drugs. So we have to go for some physical method or which the bacteria cannot develop resistance to that. So one of the approach is like the scientists observed the cicada fly wings under the electron microscope, and they observed that the cicada fly don't have any microbial growth on the surface, and they found that so it has like a nano pillar like structure, like a sharp needles, nano pillar. And if you see the size of bacteria, so most of you know the size of bacteria is in the range of micrometer. So when micrometer size bacteria fall on this nanometer size sharp needles, so it will blast, it will break. So it's like a kind of physical attack. So when you kill the bacteria by this kind of physical attack, so that bacteria cannot develop resistance to that. So it's like a kind of you are piercing the balloon with the sharp needle. So that kind of approach. So we can also make such kind of nano material based coating. For example, in our lab also we are trying to develop such kind of nano pillar structure. So with this kind of coating can be useful for the public toilets. And if you are applying this paint on these public toilets, so it won't allow the bacteria to grow, and it will prevent the several bacterial infection. So this is the one approach. So there are several approaches we can use it to overcome this AMR, that is antimicrobial resistance. And another example is if you see the butterfly. So during the rainy season, the butterfly fly here and there, and it don't get wet. So if you see that the reason is the butterfly wings are made up of like a super hydrophobic nanomaterials, and it also has a self-cleaning property. So for example, this is a super hydrophobic surface. So the red color is the dirt, and when the water falls on that, the dirt will roll and clean it. So this is a super hydrophobic surface, and also it has a self-cleaning property. So that is the advantage of this the butterfly wings. We can copy the idea, we can make such kind of product. So we can also take the idea from the lotus leaf. So most of you might have seen this lotus leaf, and if you are visiting southern side of India, like South India, like so most of the temple, so they have the small ponds. There will be lotus plant, and the prasad and other things they distribute in the lotus leaf. And also the roadside shops also they distribute the food in this lotus leaf. If you see that the lotus leaf has the nano ridges, and it is having the super hydrophobic surface, and it also has a self-cleaning property. You don't have to wash it, just dip in the water, it's completely clean. So we can copy the idea from this lotus leaf, and we can make such kind of textile material, so which will be self-cleaning. You don't have to use any detergent to wash it. You can simply dip in the water, and it will clean it. So for example, like students or somebody staying in the hostel, then no need to wash it, just dip in the water, and no need to use any detergent. So that is the advantage. So it have a super hydrophobic surface, and it can clean it. And another idea is we can take it from the woodpecker. So if you see that woodpecker nod the head at a very high speed, and again the collarbone is very strong. So we can copy the idea, and we can create such kind of car bumper, or we can also see the athletic helmets, so which will prevent the brain injury. So recently you might have observed in the IPL match also, some of the cricket players they are using this kind of neck band. So most of you misunderstood that neck band is like a kind of for headphone kind of listening the songs or something, hearing the commentary, but it's not the headphone. It is a neck band. So whenever any ball or anything hit, so it will control the blood flow to the brain, so it can prevent the brain damage. And this is also one of the successful product of the biomimicry. And not only that, we can also copy the idea from the bacteria. So most of you know the bacteria move from one location to other location by using the flagella. And if you see the size of flagella, which is in the range of 10 nanometer diameter and 10,000 nanometer long helix, and it rotates at a very very high speed, like 20,000 rpm, and within 1 milliseconds it can reverse its direction, which is not possible by any human made motor or rotor. Even if you switch off the centrifuge, or if you switch off your mixer grinder, it takes at least few seconds or few minutes to come to the zero. But it is rotating at a very very high speed, like 20,000 rpm, and it can reverse its direction within 1 milliseconds. So we can copy the idea from the bacteria flagella, and we can make such kind of nano robots or nano machine. So for example, we can have a micro camera, and we can load with the anti-cancer drug or any therapeutic material, and from outside we can monitor these nano robots. So it may look like a science fiction, but a lot of research is going on in this area to make these nano robots as like a futuristic therapeutics. So for example, if there is a cancer, like we can send these nano robots which can find out only the cancer cell and destroy only the cancer cell, and healthy cells can be saved. So not only that, in future we may not have the vaccines also, we'll have nano robots, and we can operate from outside. If there is any new virus or anything, it can go and attack and destroy it. So it has both the merits and demerits. So we have to use the right side of the any new technology. And not only that, not only for the biomedical application, we can also use it for the day-to-day application. For example, whatever the toothpaste or brush you use it, there will be always a chance for dental cavity, because it won't clean it completely. So we can, in future we may have a mouthwash which has the nano robots, so which can clean the complete teeth. So again, nowadays in the TV advertisement, they're asking whether a toothpaste is having salt or So similarly in future, they will ask you whether your mouthwash is having nano robots or not. Okay. So we got the overall idea about the nanomaterials, and we got the idea about the biomimicry. So now I'll move on to the what are the technologies we made in our lab at IIT Roorkee. So our lab name is Nano Biotechnology Lab, and again we also got the idea and interest inspired from the spider web. So we can make nanofibers. So some of the scientists, they already optimized how they can make nanofibers using different technologies. So we are more interested in the nanofibers. So in my today's talk, I'll be mainly focusing on how we can use these nanofibers for various applications. So before that, I'll briefly talk about what is nanofibers. So nanofibers are continuous fiber, and with a diameter in the range of 50 to 500 nanometer. And just to give a broad idea, if we see that this brown color is the human hair. The human hair is 80,000 nanometer, and this gray color is the nanofiber. So that much small is the nanofiber. So we can use these nanofibers for various biomedical applications. And in our lab also we are working on various applications, including antimicrobial wound dressing. In my today's talk I'll discuss about that, and I'll also discuss about tissue engineering, and I'll discuss about the drug delivery. And we also use it for water filtration and other applications, and except this ballistic protection, that is the bulletproof jacket. So we are also working on the most of the other application. So how to make these nanofibers? There are three approaches: self-assembly, phase separation, and electrospinning. So out of these three methods, the electrospinning is the simplest one and widely accepted for making the nanofibers. So what is electrospinning and what is the principle of electrospinning? If you see that the principle of electrospinning is similar to the cotton candy. So most of you might have seen this cotton candy. So the particular vendor, he put the sugar here, and the sugar will melt, and due to the high electric current, that will forms like a small thin fibers, and he used this stick and collect these fibers. So basically what we are eating is the microfibers. So we are using this cotton candy machine, and we are making these fibers. The principle of electrospinning is almost similar. So here we are going to use a syringe, and in the syringe we are going to add the polymer. Instead of adding the sugar, we are going to add the polymer, and instead of using the pink color, we are going to add any FDA approved anti-cancer drug or any therapeutic material. So once you apply this high electric current, what will happen is that due to the electrostatic potential applied between the spinneret and collector, so this is the collector, so this is a stationary collector, and this is called a spinneret. So when you apply this, what will happen is that the particular viscous polymer will come out through the syringe, and due to the accumulation of charge, so this will take like a cone shape. So this is called a Taylor cone. So that is named after the scientist Taylor, and due to the more accumulation charge and this thing, so it forms like a fiber. So that fibers can be collected in this grounded collector, and the distance between the syringe and the collector is very, very important. So if you are keeping the collector close to the syringe, you get the microfiber, and if you are keeping it the optimum distance, you get the nanofiber. So and again, you are keeping the collector further down, so you get the broken fibers. You don't get the pico fibers, you get the broken fibers. And the viscosity of the polymer is also very, very important. If it is too much viscous, it cannot come out of the syringe. If it is very less viscous, instead of forming fiber, it forms like a beads. So we have to optimize the viscosity, we have to optimize the distance between the syringe and collector, and we have to optimize the particular voltage, and we have to select the right combination of polymer and right cross-linking agent, so that the fiber will be more stable and strong. And this is a rotating drum collector. So the previous one is, I told, stationary collector, and this is the rotating drum collector. And so here this is a horizontal setup. So the fibers will be coiled like the rotating drum. Okay, it will rotate in this direction. So all the fibers will be coiled on the particular rotating drum. It will be like a kind of thread bundle. So what is the advantage of using these stationary collector and rotating drum collector? When you use the stationary collector, you get this kind of random fibers. So, and it depends on your application, you can select the stationary collector, and when you use the rotating drum collector, you get the proper aligned fibers, So most of the neurobiological studies and microbiology studies, so we use this rotating drum collector to get the aligned fibers, and most of the wound dressings and drug delivery we can use the random fibers. So depends on your application, you have to select the particular collector. So now I'll discuss about the different applications. The first one is how we can use these nanofibers for anti-cancer applications. So here we use the core shell nanofibers. So the core shell is like we can have the nanofibers. We can make nanofibers core and shell. So it is like a kind of pen and refill. So the advantage is inside we can load one type of drug, and in the shell we can load another type of drug. So for example, here we loaded one type of drug, and in the shell we loaded another type of anti-cancer drug. So when you loaded two types of anti-cancer drug, so we can overcome the anti-cancer resistance by the cancer cell. So that is one advantage. And also when you combine two different types of anti-cancer drug, it will have a synergistic therapeutic effect. And you can also observe here we made like a line, that means it is a cross link, and here so we did not do any cross linking. So if you are not doing cross linking, the polymer will melt faster and it can release the drug very faster. So we can control the drug release also. For example, in this case the drug release will be very slow because core as well as shell both are cross linked, and here the core is not cross linked and only the shell is cross linked. So we can also fine tune or control, play with the drug release also. And this is how the final product look like. So this is the anti-cancer drug loaded nanofibers, and again, so this is loaded with the anti-cancer drug curcumin. That's why it just look like a yellow color, and you can see this in this animation. So these thread-like structures, these are nanofibers, and you can see that this cancer cell which is longer, and once you see that like the drug is released from this nanofiber and it become rounded. So the cancer cells, once it undergo the apoptosis, it become rounded. So the nanofiber is releasing the anti-cancer drug and it is killing the cancer cell, and it's finally it become a rounded shape. And the core shell nanofibers provide a control and sustained release of this anti-cancer drug, so that can prevent the local tumor reoccurrence. Because if you see that like whenever the doctor do the surgery, they have to remove the tumor completely. So if they're leaving even a trace amount of cancer cells by mistake, so that will regrow and create problem. Then again the patient has to go for surgery. So whenever the doctor do the surgery, when they remove the tumor, so what they can do is they can use this bio bandage, and it can release this anti-cancer drug, and it can kill the cells which is a reoccurrence due to the leftover cancer cells, and also it acts as a kind of physical barrier, and it can prevent the metastasis. So this way we can have overcome the post-surgical reoccurrence of tumor. And you can see that this is a control, that is the untreated one, and whereas when you treat with the anti-cancer drug, it is undergoing the apoptosis, that is a natural cell death process, and the cells become rounded. And this was highlighted in Nature India. And the next application is like we are going for like how we can use these nanofibers for wound dressing application. So again we can also use different kind of materials for wound dressing, but here we selected silk as a material because it's a biomaterial and also FDA approved. So using the silk we made a bio bandage. So most of you might have observed, like whenever we get a wound, we put the bandage. The main painful is when we remove the bandage, it's very painful, and also it damages the wound. And again, for diabetic people, the getting wound healing is really challenging. So what we have done is like we made a multi-layer, that 3-layer, triple layer nanofibrous bandage, and for that we use the silk fibroin and silk sericin. So the silk fibroin and silk sericin, we took it from the silk cocoons, we purified, and if you see that this silk sericin is okay, so silk fibroin is hydrophobic in nature, so this will be the top layer, and silk sericin is hydrophilic, so that is the middle layer loaded with the drug, and bottom layer again we use the silk fibroin. So it act like kind of similar to the commercial bandage. The top layer is hydrophobic, so it won't allow the waters to enter, and again it slowly degrade. So we can again play with the cross-linking agent, and we can increase the or decrease the porosity, and we can control the drug release, and also we can control the degradation rate. So when we did the animal study, we found that like when compared to the commercial bandage, the commercial bandage took 16 days to heal the wound, and our bandage within 12 days it is able to heal the wound, and also there is a proper hair growth, there is no scar formation. So it's like a kind of natural healing process, because the bandage is made up of biopolymer, and again we don't have to remove the bandage, it will be degraded in the wound itself, and also it favors the cell attachment and cell growth, so the healing is faster. So that way it can heal it. So presently we are also working on developing into several other products. For example, we are making the nano gel and hydrogel based products, so that so we don't have to even make this bandage, we can apply this nano gel or hydrogel. And one of my another student, she is making the liquid bandage, so instead of making like a gel, we can apply this liquid, and which will solidify in the wound, and again it will also degrade with respect to time. So the third application is how we can use this nanofibers for tissue engineering. So what is tissue engineering? So most of you know the genetic engineering means modifying the genetic material, and tissue engineering means we are making artificial tissue in the lab. So why we have to make artificial tissue? Because there is a scarcity for the organ donor. Whenever there is an organ damage, so we have to depend on organ transplantation. So for organ transplantation, the tissue should match. Even some of your relative or some of your friends are ready to donate the organ, but the organ should match. And again, even if it is matched, so we have to under several other drugs treatment, so that the organ will stay in your body for more number of years. So that is one of the major challenge. So in future we have to depend on the artificial organs and artificial tissues, and for that we can use the nanofiber as a kind of support, like a kind of it can be useful for making the scaffold for artificial tissues. So if you see the statistics in US, if you see that like roughly 1,19,000 people need the organ transplantation, but only 30,000 people are able to get the organ transplantation, and each day at least 22 people are dying due to lack of suitable organ transplantation. So in US alone, if you see that 22 people are dying, you can see the whole world how many people are dying due to lack of suitable organ transplantation. And whenever the organ is functional, we don't know the value of the particular organ. And if you see that the cost of this each and every organ, you can see here. So again, it is not the kind of approved rate or anything. This is again the black market rate which published in this particular newspaper. So you can see here the cost of each and every organ. These are in the pounds. You have to multiply by 100. So you can see that how much worth it is. Even while teaching to my students, I usually say that like whenever your parents scold you like useless or worthless, you can show this slide again how much worth you are. But the problem is even your parents may again say that don't ask any property. I already given this much worth property, so it can also backfire. So this much worth each and every organ cost. Okay, so the cost of organ is very very expensive. Like if we know the value, like we don't misuse any organs, and if it is get damaged, it's very difficult to get these organs. So lot of research is going on. So one of the research is again to make the suitable scaffold, to make a three-dimensional scaffold, because usually we grow the cells in the two-dimensional cell culture plate, so which is not like your original organ. So organs are mostly three-dimensional, so we need a three-dimensional support, like a foundation for the building. So initially scientists used micropore scaffold. So the drawback is your cell is also in the range of micrometer, so there are a lot of gaps. It does not mimic like your original tissue. Then they moved to microfiber. Again the same problem. The cells are growing only on the particular the supporting axis, not on the complete. And then they finally use the nanofiber scaffold. The advantage is the nanofibers are in the range of nanometer, and your cell is in the range of micrometer. So it exactly mimic like your original ECM, that is the extracellular matrix. So if you see any tissue, extracellular matrix is act like a kind of foundation or scaffold, and on the top of that you have the micrometer size cells which will attach and nicely grow. So it mimic like your original ECM. So you can see this is a single cell, and it is contacting thousands of nanofibers. So the single cell can contact thousands of nanofibers, so it can transmit the fine signals, and it closely mimic like your extracellular matrix. So it can be like a kind of useful for making the scaffold for making the artificial tissue. And so we also use the nanofibers for making these artificial tissues. So, and we name it as a kind of 4D scaffold. So whatever the scaffold available in the commercial market, so as I told you, like most of the lab, they use this kind of glass or plastic plates for growing the cells, which is two-dimensional, which does not mimic your in vivo conditions. And commercially we have a three-dimensional scaffold. Again, the three-dimensional scaffold is static. Okay, so we don't have any dynamic scaffold. So that's what we made a dynamic scaffold, and we named it as 4D scaffold. So we can make a scaffold which can shrink and which can expand with respect to the external stimuli. So you make a 3D scaffold and which will respond with respect to the external stimuli, then it is called as 4D scaffold. And again, if the scaffolds can rotate, then it is called as 5D scaffold. And when you combine this 4D and 5D, that is called as 6D scaffold. So it can respond with response to the external stimuli, and it can also rotate, then it is called as 6D scaffold. So what we did is we made a multi-layer nanofiber scaffold. So it is like a 3D scaffold, but it can respond to the external stimuli. On both the sides we added the magnetic nanoparticles. So when you add the magnetic nanoparticles on both the sides, and with the help of external magnet, we can make this scaffold to shrink and expand. So this is a kind of dynamic scaffold. Usually normal scaffold, commercially available scaffold is 3D, which is static, which cannot shrink or expand. So why we need the scaffold which can shrink and expand? For example, if you want to make an artificial heart or artificial lungs, and you want to understand their behavior in this in vitro condition, so this scaffold will be helpful. So you can seed the lung cells or heart cells in the middle of this, and with the help of this external magnetic field, we can make the scaffold to shrink or expand. So based on that, so this is kind of artificial gym for the cells. We can mechanically stimulate the cells and understand so whether the cells can withstand the particular mechanical load. For example, you are making some artificial tissue for repairing the lung damage or repairing the heart damage. So if you are growing the cells in the normal static 3D or 2D, when you put it into the patient body, suddenly due to the mechanical load, it cannot withstand, and the success rate of transplantation will be low. In addition to that, what we can do is we can use this 4D scaffold, and we can train the cells. That means we are exposed to such kind of mechanical stimuli so that the cells are strong when you do the transplantation, and which can adapt to the environment immediately. So for example, you can see here, so when you stimulate these cells with the mechanical stimuli, and you can see that when compared to the control, after the cycle 3, you can see that number of like gene expression and protein expression is very higher with respect to the number of cycles. So the simple layman example is, you can see that like for example, the two boys staying in the same hostel, eating the same hostel food, the person who goes to the gym, he get very good muscular development. So the reason is he is giving the mechanical stimuli. So when you do the exercise, what will happen is the mechanical signal will induce the gene expression, then it will induce the protein expression. So that's why the muscular development is happening. So that is called as mechanotransduction. Mechanical signal became a chemical signal, mechanotransduction. So the particular field is called as mechanobiology or biomechanics. So we can use this kind of mechanical stimuli, and we can induce the mechanical stimuli, and we can understand what are the genes are expressed. So it can be a kind of artificial gym for the cells. Like before we transplant the heart cells or lung cells, we can train the cells, and it can have more success rate. And when you stimulate the cells, what will happen? The cells have like a finger-like structure that is called the phylopodium. The cells moves in the direction, and it uses the finger-like structure, the phylopodium, and these actin fibers combine and provide the strength. And when we used our aligned nanofibers, and this is the cell and these are the nanofibers, and when you give the mechanical stimuli to the cell, so the mechanical stimuli is similar to the roller coaster ride. So when you go roller coaster ride, we grab our mother hand or sister hand very tightly. So it is similar to that. When you give the mechanical stimuli to the cell is grabbing these nanofibers, and if you see the displacement, it is close to around 5 to 10 micrometer displacement with respect to the mechanical stimuli. So we can use as a kind of model system for understanding the role of mechanical force in the cell biology. So what is the like a mechanical field role, and we can understand the role of mechanical forces, and we can develop such kind of tissue which can be exposed to the mechanical forces before we transplant the tissues. And this work was highlighted in the ACS Applied Biomaterials cover page, and also we received this BIRAC Gandhi Neng Technology Innovation Award to make it into final product from the Vice President of India at that time, Dr. Venkaiah Naidu. And we also developed this product into the more mechanical biological device, and recently we also published in a small journal, and it is also like the device is also finally be patented, so which is giving a very good results. And in future, so the tissue engineering, we have to again grow the tissues we have to make this nanofiber scaffold, and we have to grow the tissues. So again, so which is a time consuming process. So the future is moving towards a 3D bioprinter, that is we can print the organ. So similar to your normal inkjet printer, instead of using the color ink, we can use the bio ink. So the bio ink is a combination of protein and cells. So when you use this bio ink, and we can print the particular organ or tissues, and for example in the normal printer we need a paper. Similarly, instead of using the normal paper, we'll be using this bio paper. So the bio paper, mostly it will be like a temperature sensitive or pH sensitive. So we can use this bio paper, and we can print it like a kind of layer by layer. So we can put the cells and we can put the cells layer by layer, and once the printing is done, the cells are fused, we can remove before the cells are fused, we can remove the bio paper. The simple layman example is like whenever we construct our home or the building, so we use the bamboo as a support for the roof. So once the roof is stable, then we remove the bamboo, so the roof will be stable. Similarly, we use the bio paper as a supporting material scaffold. On the top of that, the cells will be like a small spheroids will be printed layer by layer. Then we can remove the bio paper by simply changing the temperature or pH. So in this way we can make the complete 3D printed tissues. But one of the major challenge with this 3D bioprinter is like we can print the heart or kidney, but the problem is to make it more functional. To make it more functional, we need a lot of blood vessels and multiple cell type. So that is one of the challenges scientists are facing. So still the lot of research is going on in how to print the blood vessels and make it more close to your original organ, and how to make the heart to beat. For example, again it needs the help of electronics people. So we need similar to the pacemaker, we need some embedded nanofiber membrane, and which can work like a pacemaker and make the heart to beat. So we can print the heart, but we need the help of electronics engineer to make the heart to beat. But people are successful with the artificial nose and ear, but again it is like a kind of like it can stay for a few years, like it's artificial nose and line. So a lot of research has been done with some of the skin, artificial skin and other organs are made, but the other organs like lung and liver and kidney, it's very difficult to make. So not only for the biomedical applications, we can also use it for making the burger, the test tube burger. It's a completely lab made artificial meat. So we don't have to kill the animals to get the tissues. So that's called like for example during 2020, during the COVID time, Singapore has approved the artificial meat for human consumption. Like, so we can use these cells, and we can grow the cells, and we can use the bioprinter, and we can print it, and we can make the completely artificial meat, and without the need for killing the animals. So how we can make it? We can take the particular stem cells or the cells from the particular animal and grow the cells in the lab in the 2-dimensional environment. Then you grow it in the 3-dimensional environment. Again, if the meat has to taste like your original meat, it has to undergo the same kind of mechanical. As I told you, this role of mechanical force is very, very important. So we can also exercise the muscle to boost the protein. So when you use the artificial stuff to that, as I told you, like our 4D scaffold, we can similar kind of scaffold can be used to make the cells to do the artificial like a gym. So it can do the so that such kind of protein will be synthesized or expressed. Otherwise, the meat won't taste like your original meat. So that's what whatever the animal exposed. For example, if you are eating the leg part of the particular animal, so it has to bear the such kind of weight load and whatever the physical mechanical load it has to be exposed. We have to provide an artificial environment. So then it will produce a particular protein, and it will taste like a original meat. And again, we can also add the flavors, and again we can fortify the meat enriched with vitamins and these minerals, and again, so we don't have to kill the animals, so we can make the artificial meat in the lab and we can ready to eat. And again, another new concept is instead of again going for this animal based meat, so another concept is like a kind of vegan meat. So it is also popular in Singapore and other countries. Like it is again mainly based on the vegetables. Like we can use the jackfruit powder, and again they use the beetroot extract, which the color will be look like your the blood. So we can use the beetroot extract, and we can use the jackfruit, which will taste like the meat, and they are making this completely plant-based meat. So that is also very popular, and you cannot find out that it's a real meat or the plant-based meat. So the taste is almost similar. So that is also one of the upcoming topic. Like if anyone interested in the research, you can also explore it. So there are some 3D bioprinters are available for plant-based meat also. And during our COVID time, we also use this nanofiber mask. And what we did is similar to the N95 mask, we use the normal, that green color 5 rupees mask. And if you open this, the 5 rupees mask, in the middle area there is a space. So where we can put our nanofiber membrane, and the cost wise it is less than 5 rupees, and the efficiency wise it is equal to the your N95 mask. And again, it has nano silver and nano graphene, so it has antibacterial, so we can use it several times. It don't create any odor or any smell. At the same time, it has a graphene. It can also use during the pollution, like it can absorb the smoke and other polluted smoke. So that is the advantage. So in our lab also we have the 3D bioprinter and 3D food printer and electrospinning setup. So whenever if anyone wish to come for using this facility, you can also always. And again, we also use this nanofiber for making the edible coating and packaging. So one of the major challenge in the nowadays is you can see that the fruits and vegetables, like especially this apple, it is very shiny in nature. And again, if you see that most of the apples comes from Australia, New Zealand, and USA, it comes with the sticker, and it stays for another 20-30 days in the ship from that country to India. Then again it stays in the market for 20-30 days. Again it comes to your room and home, again 10 to 20 days it stays. So but it stays fresh. So there are a lot of things are modified at the genetic level also. Again, another thing is they also do wax coating. So what happens is when you regularly consume this wax coated apple, so it leads to the cancer. So instead of that, what we can do is we can also go for edible coating, that is the biocompatible coating. So that's what we use, the silk based nanofibers. The silk based nanofibers, again it's FDA approved. Again, if you eat, even with the, suppose when you wash it, the coating will be removed. Even if you're eating little bit trace amount of this silk also, so it will give additional protein to your body. So this way we can have a kind of particular packaging, nanofiber based packaging. So we made a silk based biomaterial, and we added this haldi, that is a curcumin, for antioxidant and antimicrobial property, and honey we use it for moisture absorber. And we demonstrated with the model fruit, that is banana, because the Asian countries export banana and it gets spoiled. So if you see that, like the banana, this uncoated is spoiled after 10 days, whereas the coated one is stays fresh even after 10 days. So that is the advantage of this nanofiber based coating. And when I presented this work in one of the conference, so another person raised a question, like it is made up of silk protein, that is again a kind of insect protein. So the vegetarian people may not be comfortable. So then we got the another idea. So we made a plant based coating. So again, the plant based coating, we taken the idea from our home kitchen, that is like a pickle. So the pickle stays for a longer time and it stays. So the pickle is made up of tamarind and vinegar. So similarly, we made 2-3 combinations, and we optimized the plant-based edible coating. So the advantage is when you use the plant-based edible coating, so it is like a liquid form. Simply you can dip it and dry it. So it will increase the shelf life of the fruits. And also the additional advantage is the tamarind has the property to uh, degrade the pesticides. So nowadays the pesticide is a major problem. Like most of the fruits, vegetables comes with the pesticide. So the tamarind has the property to degrade the pesticides. So when you wash the coating, along with the coating, the pesticides are also removed from the fruits and vegetables. It is kind of eating the equivalent to the organic fruits or vegetables. So that is the advantage. So with this slide, I'll conclude my talk. There are several other applications we made it. So, but I restricted to the nanofibers. So we also made several nano devices for cancer sensor. So we also made a small nano device similar to the blood glucose measuring device. So we can also diagnose the cancer at the early stage. We also file a patent and technology is ready. So similar to the blood glucose, where you put a drop of blood and measure the blood glucose, we made a device where we can put the drop of saliva and we can find out whether the particular person is having the oral cancer or lung cancer. So that device is now we made it prototype. We are going for a clinical trial. So again, so that is slightly different from this topic. So I just now discuss about that topic. So we are mainly working on different biomedical applications of nanomaterials, and this is my nanobiotechnology research group. And I thank all the funding agency and all the students who supported. And we also have a startup company that is Super Good Nano Private Limited. We also make some products, and I also teach 2 courses under the NPTEL scheme. Like one is biomedical nanotechnology. This is mainly in the autumn semester, and this one is in the spring semester. So if anyone interested, the registration is open for this course. And this course is a theoretical course, various biomedical applications of nanomaterials. And this is an experimental course. I have shown the video demonstration of how to make nanoparticles, how to characterize the nanomaterials using different techniques. So also the troubleshooting and making the nanomaterials, all the things are discussed in this course. And also the lectures are available in the YouTube also. If you don't want to register for this course, you can also go through the YouTube videos, and we can also suggest to your students. And additionally, recently this NPTEL has introduced a new scheme that is called NPTEL Pre-Doc Fellowship. So under this Pre-Doc Fellowship, for example, suppose someone want to do research at IIT, not only IIT Roorkee, any IIT. So for that, you have to take at least 3 NPTEL courses. So you can take 3 NPTEL courses, and you have to score at least 60%, and you have to also submit all the assignments on time within the deadline, and you have to write the exam and score 60% in 3 courses. So then you get the opportunity to do the NPTEL pre-doc fellowship. That means after completing your BTech or MTech, or it can be after PhD, or somebody, even some faculty want to do for 1 year training at IIT Roorkee or any other IIT, so that is possible. So after that you have to apply for this fellowship. So monthly stipend will be 25,000 and 10% HRA, and also they also give 1 lakh for the chemicals for your research work. So last year also it was it was introduced last year. Last year one student joined with me, and so before joining this fellowship, he was not qualified for the GATE. So when he was doing project in my lab, he qualified the GATE exam, and now he joined the PhD with me. So it will be a good opportunity for the students, so they can do this course and come to IITs and do 1 year research work, and they can publish some work. They can go to abroad for PhD, or if they qualify GATE or any national level exam, they can also join PhD in IIT. So that is the advantage of this course. And this NPTEL also give a different kind of badges. Like if you are submitting the assignments on time, they give NPTEL Discipline Star. If you are scoring 60%, they give particular star type. And if you are taking more number of courses, they give NPTEL Superstar or Megastar. So any one you can qualify, and that can be useful for making your NPTEL pre-doc fellowship eligible. So then you can select any IIT nearby your institute, and you can do the particular project. Okay, so I'll end my lecture with this slide, and thank you all. And this is our institute, IIT Roorkee, so which is more than 178 years old, and this building is more than 178 years old building. So whenever you get chance to visit IIT Roorkee, so drop a mail. We can come and visit the not only the Roorkee campus, and also you can visit our lab, and you can see the facilities. Okay. So thank you all. If you have any questions, I'll be happy to answer your queries.
Thank you, sir, for such a valuable and informative session. Um, Now I invite participants to share their question in the chat box. We have one question. How do we determine the optimal concentration of nanofiber coatings in horticulture applications? Is it different from different horticulture crop or same?
Yeah, it is depends on your the particular fruits or vegetables. Like we have to optimize. There's for example, the strawberry, it goes bad very faster. So it depends on the fruits, depends on the nature of the fruits. We have to optimize the thickness. We have to optimize the particular polymer coating also.
So I have one question.
Yeah.
So in electrospinning, like what is the temperature, maximum temperature? Like how do we take care of that the biomolecule does not get degraded while we are applying potential voltage?
Yeah, when
So
you apply
like,
the potential voltage, the temperature won't rise. Like we have to again the temperature also we have to optimize. Now accordingly we have to select the right choice of polymer. So the temperature will be like optimal room temperature, so that can be controlled. So in this way we can make sure that the drug is most of the drugs are like not temperature sensitive. So if it is very sensitive drugs, it will be challenging. But when we tried with the different drugs and the DNA, it is highly stable. So we use with different polymers, it is optimized, so it is stable in the different parameters.
no, like protein, like for example, if there is a therapeutic protein, so proteins are susceptible to degradability, or is it not in electrospinning?
Yeah, protein, the silk, it is a protein, but again, the
Yeah.
silk protein has very good temperature, thermostable. Even you can autoclave it also. After nanofiber, you can do the
Okay.
autoclave. It's stable even up to 120. So it depends on protein. Some proteins are very sensitive. So for that, we have to optimize the ratio with the polymer. We cannot use it directly the protein. We have to mix with the some polymer so that we can reduce the degradation of the particular protein. But some protein like silk,
Yeah.
this is highly thermostable. So we also done
Okay.
autoclave also after this. So it is very
So
stable.
silk fibroin you extracted from silkworms, or it is was it commercially available?
It is commercially available also, and
Okay.
we extracted from the silk cocoon. Silk cocoon is a waste of the silk industry, and
Mm-hmm.
there's a simple procedure. It's like a heating and degumming procedure. So we can extract the different protein also.
Yeah. So any other question we have? I think, sir, there are no other questions in the chat box.
Okay.
Yeah. And thank you so much, sir, for accepting our invite to be the resource person for this FDP and enlightening us with starting from basics to how technologies are developed for various applications. Thank you
Okay.
so much.
Okay. Thank you very much. Thank you for that.
Thank you. Thank you, sir. So our next session will be at 7:30. I'll just post the attendance form. You all can fill it. And at 7:30, we'll be joined by Professor Mitali Mukherjee from IIT Jodhpur. She'll be talking upon on personalized medicine, intelligent health applications for personalized medicine. Uh, Thank you, everyone. And I'm just posting the attendance link. The meeting is on. We'll get back at 7:30. Ma'am, we can start the session now and we'll announce this thing.
Okay.
Yeah.
Are
So
you able to open it? The survey, can you just try?
let me check, ma'am. Yes, ma'am, we can fill also.
Okay, so if people can fill this form a little bit, then it'll be fun. So
I'll start myself. Let me just finish it and then we'll start.
yeah, we can tell the others also to fill.
Yeah, ma'am. I'll put it in the chat box. Kindly fill the form. Ma'am, I have filled the form.
Yeah, Phil, yeah. I just
Anyways, I'm also an Ayurveda enthusiastic, so
okay,
I believe in this form of
okay,
medicine. Yeah, I have filled the form. So I
great.
request everyone, all the participants, to fill the Google form which ma'am has shared. So this is just a quick survey which she wants from us.
In the meanwhile, we can start also.
Yeah, yeah. And once you have filled the form, you can put it in the chat box that you have filled the form. So now we'll start the session, ma'am. Good evening. And we'll start with our session 2. Professor Mitali Mukherjee from IIT Jodhpur has kindly consented to give talk on intelligent health, precision health, personalized medicine. Now I request Twinkle to kindly introduce our eminent speaker. Twinkle, are you there?
Yes, ma'am. Thank you, ma'am. I am pleased to introduce our speaker, Professor Mitali Mukherjee. She is a professor and head of Department of Bioscience and Bioengineering at IIT Jodhpur and also affiliated to the School of Artificial Intelligence and Data Science. Before joining IIT in May 2021, she was a chief scientist at CSIR Institute of Genomics and Integrative Biology, Delhi, and headed the Department of Genomics and Molecular Medicine. Her broad area of research interest is in understanding human genome structure and variation for its application in the discovery of biomarker for predicting susceptibility to diseases, understanding human phenotypes and environmental adaptation, and mapping mutational histories. She has been involved in setting up key genomics initiatives in the area of rare genetic disorders, population genomics, functional genomic of repetitive sequence, and initiated a new area of research, genomics, in the country for integrative medicine. She was the convener of the Indian Genome Variation Consortium project that provided the first comprehensive genetic landscape of the Indian populations. Led the IGVC project has been used for tracing human migration and history of diseases, identifying founders in different hereditary ataxis, signature of human adaptations with respect to geoclimatic conditions and pathogen prevalence, and developing genome-based diagnostic primarily in the area of neurodegenerative disorders. She also led the Indian team for HUGO Pan-Asian project, which provide insight to peopling of Asia and brought out relatedness of Indian population with other Asian population. At IIT, she is currently involved in developing and coordinating two major transdisciplinary initiatives, HAR Design and Center of Excellence in Integrative Precision Health. He is elected Fellow of National Academy of Sciences, Fellow of Indian Academy of Sciences. She is recipient of several awards like Kirtan Sanjeevani Pushpalata Ranade National Award for Women in Research, Vasvik Award for Women Sciences, Pandit Shiv Nadar Sharma Shod Puruskar for Research in Ayurveda, Shanti Sarup Bhatnagar Award in Medical University, National DBT Young Women's Bioscientist, etc. With that, I now request Professor Mitali to proceed with the session. Over to you, ma'am. Thank you.
Thank you so much. Thank you for inviting me for this session. I love to talk about this subject, and I hope I do enough justice. So we will try to have question answer session after the talk to be able to understand whether how did you find the lecture, and whether if you have any other questions from there. So I hope you all would have filled a questionnaire. This is just a quick survey. Basically, you're trying to assess your awareness of Ayurveda. But let me start the lecture. So, just a quick thing: is my slide visible? Can somebody confirm?
Yes, ma'am.
Yes,
Yes,
ma'am,
ma'am.
it is visible.
Okay. Okay. So this is IIT Jodhpur. It's a very beautiful campus. I am at present here. I spent 23 years in Delhi, and then I moved 4 years back here. And I'm going to share to you something of a longer journey of 25 years in a particular area of research, which I think could offer intelligent health solutions to the challenges of the present times. Uh, the present time, all of you would have heard about One Health framework, where people are now trying to understand how to kind of work towards a comprehensive understanding of animal health, human health, and environmental health, which kind of is uh, governing a large meaning. We need to understand it comprehensively because of the transitions in terms of climate, pollution, deforestation, biodiversity loss, people moving from one part of the country to the other, social structures are changing, dietary transitions are there, antibiotic and antimicrobial resistance, emerging problems of disease vectors, and also many other kinds of intensive livestock farming, which is leading also to many other problems. And if we want to kind of tackle all these challenges, because they are interrelated: what we eat, how we grow plants, how we take care of our livestock, what gets into the circulation, and then that causes what kind of health hazards. All of the things are interrelated, and we really need a transdisciplinary framework to be able to address all these challenges in a comprehensive manner. So the need of the hour is how we can make healthcare affordable, how we can make it scalable, how we can have precision and early actionable interventions, and how do we do that in the context of the immense diversity of our country, and also other kinds of demographic aspects, and how do you ensure sustainability. So we are trying to develop some frameworks which are towards addressing this. So, as especially because now I'm residing in this desert eco region, which is also a unique eco region where everything is in the extremes of temperature, scanty vegetation, aridity. So, there are certain kinds of challenges over here. So, we are trying to understand whatever frameworks we are in the context of the desert, and also an IUTech project for integrative medicine. So when it comes to the health challenges, because I primarily focus on health, disease, population, human population, so I will be dealing the lecture around these lines today. So there was a study in which was published by WHO in 2017, where they studied around 354 diseases across 195 countries, and it was to look at the global burden of diseases and across age group. One of the things which came out very strikingly is that we've been able to reduce substantially infectious diseases over the last many years, and so the longevity overall has increased. But what has actually happened is the last 10 years of our life we live with one or the other complex disease. So with that, the health longevity has reduced, and major fraction of that is communicable non-communicable diseases. There's this interesting book we all would have heard or read, Survival of the Fittest. This is an interesting take on that. It's called a Survival of the Sickest. So this book primarily kind of questions if there is this phenomena of survival of the fittest, then why are so many people sick, or why do genetic makeup of certain genes uh, are still there and continues to contribute to the burden of diseases if they are associated with sickness. And it actually through many compelling examples show that there are some environment or some conditions where the selection pressures are such that it allows us to have certain genetic versions which give us an advantage. But then if we are transitioning to a new environment, those very same genes might actually be not keeping us healthy. So for example, if you look at African countries, then people tend to, because of intense heat and all, they would lose a lot of salt through perspiration. So there is a variation in a solute carrier gene which allows them to retain salt. But then what happens when these people move to higher latitudes and they don't sweat so much, which is the situation in Afro Americans. So it turns out that when you are not sweating and you have a copy of the gene which ancestrally allowed you to retain salt, then that itself makes you more hyper predisposed to hypertension in comparison to the relatives who are residing in the native regions. So this is the book which provides many such examples, and it also highlights how we have to contextualize the variations in the in light of epidemiology, in light of other kinds of aspects, and same gene, same genetic variation responsible for a disease might not be so for every part of the world. The other thing is that now people are talking about precision medicine. The idea is that supposing we were to know what are the genetic burdens or genetic defects is there in the prenatal stage, and one can do a testing so that you can plan a baby when the fetus is born. Then, or when there's a newborn, sometimes there are some kinds of inborn errors which can be rectified by doing a genetic testing. Then there are certain things which predispose you to disease. So if you know that undiagnosed disease, then maybe some kind of preventions can be taken into account. Or if there's infectious disease, and not every person is equally susceptible. So who are the people who are more at risk of the disease? So could you understand that when there's cancer and there's a precision therapy, how do you identify who would respond to what kind of therapy? Or medicine, it's called pharmacogenomics. uh, and then how do you increase the health span? And now people are also talking about molecular autopsy, that if you were to understand why people die, then through analyzing the genomes, then you might be able to understand what causes death, or what other how to prevent that. So so from pre-womb to tomb, now people are thinking about individualized solutions, and maybe the genome study of the genome or our genetic makeup might be able to tell us about it. But then there are other kinds of things which people are now observing, that typically when there is a drug trial and a drug comes to the market, it comes out of clinical trial, randomized controlled trial. But then and that's population based. But when we go on to use it on an individual, not every individual is equally benefited from that medicine. So there's something called number needed to treat, which has come out of long big trials on people. And it actually tells you that supposing there's a medicine, omeprazole, for heartburn, you give to so many people, there's one blue and so many red. So this is the blue, the number of reds and the how many blues are there is something which tells you how effective the medicine is likely to be. So if there are five individuals and only one has an effective outcome, the NNT is 5. And if you have 20 and you have one, then it is NNT is 20. So the higher the value of number needed to treat, the less effective is the medicine in an individual. And so now people are trying to think that if there is so much individual variability in response to drug, then maybe today's trial designs have to have a rethink, where an individual becomes the case and control, and it's called as n of 1 trial, so that you understand in an individualistic manner who are the people who are likely to respond in what way, and you aggregate such kind of an information, and then you know how do you stratify the medicine in such a manner that most of the people are effectively treated. There are other kinds of things. Now people are also understanding that we have one thing as genetic predisposition, and the other thing is the kind of different kinds of exposures which make us which modify the course of the disease. And each of us might have a different trajectory to the health, depending on what kind of triggers we are faced with, and what kind of genetic makeup we have to respond to those triggers, like how resilient we are, how susceptible we are. That will determine final outcome of the health trajectory in an individual. And even in a disease condition, for example sickle cell anemia, many people, most of you would have heard about this mutation and this disease. 99% of the people have the same mutation, so it's not the diagnosis of the disease. But what happens is sickle cell patients progress differently to different kinds of endpoints. Like some people might have stroke, some people might have intense pain, some people might have hemolytic crisis, and this is exacerbated in the presence of certain kinds of environment or certain kinds of modifiers. So as a result of which, the problem is not with sickle cell, but the problem is how do you kind of understand who are the people who are likely to get these outcome, and is it dependent on a person's genetic makeup, or is it all random? So these are the kinds of questions, because how do you manage a person's quality of life who has got sickle cell anemia becomes important to understand. So there is gap in knowledge, that if you have so many individuals, there are individuals who are of different kinds, and might have different risks. So how you can stratify the individuals in such a manner that supposing you are giving a medicine, and if only one person is benefited, then can you identify that group from the other group, so that you can reduce the number needed to treat, and you do not harm many people by the side effects of the disease. So such kind of thought about individuality, about precision medicine, which now at this point of time we are delving into, is not new to India. So 5,000 years back, maybe the timing are not really that much documented, but this is what is generally thought. We had this system of medicine which actually has in its tenet individualization. So this is acknowledged now widely, and this Journal of Nature Outlook, which dealt with precision medicine, actually talked about in India the concept of Ayurvedic medicines are laid out in Vedas, and the goal is to maintain balance between body, mind, and spirit. And treatments are tailored to person's constitution. And if you look at the Charak, uh, this is Charak Sutrasthan, so this is the reference. The English version of that reads as aim of Ayurveda is maintaining health in healthy and alleviation of disorders in diseased. So you can look at healthy and diseased connotation, which itself says individualization. So they are not talking about population, but they are talking about the individual, and how do you maintain health in an individual, or how do you alleviate disorder in a diseased, is what the aim of Ayurveda was described as. And also, when during COVID, we all felt that it's a new and emerging disease, right? But in Ayurveda, such kind of diseases as pandemic are described as Janpad Udhvans. And if you read the version of what they describe as pandemic, it would read as a WHO kind of a definition, where it talks about even if there is dissimilarity in the physical constitution of human, there are factors which are common to individuals, and vitiation leads to simultaneous manifestation of disease having same set of symptoms leading to destruction of countries. And these factors are air, water, location, and season. This is what is given vividly, and this is documented thousands of years back. So when it came to COVID, it was a great leveler because many people availed COVID either as a way, Ayurveda as a way of prevention, as a way of cure. In many, and this is such a kind of survey I've done in many forums. So if when I do 1,000 survey, then we can see a fraction took it during treatment, large fraction in prevention, many in both cases, and one third of the fraction did not use Ayurveda. So COVID pandemic is was a great leveler because for Ayurveda, any disease, not only COVID, is business. COVID was a business as usual because every disease is today described in modern terms, and to find an equivalent term in Ayurveda, you need to understand it from the foundational principles of Ayurveda. And the fact that uh, people are still prescribing, and there is a whole market which was 16,230 million and is expected to increase by 12%, tells that if so many people are buying, then if definitely some people are benefiting. And to be able to understand that in a kind of a unbiased manner, this was one of the studies which was undertaken by using digital data captured in Ayurveda settings of Jiva Ayurveda. So this was the first data driven view of Ayurveda practice. It recorded digital data of 53,000 patients by 300 Ayurveda doctors who are involved in teleconsultation. And this alluvial plot actually shows that in which kind of diseases Ayurveda medicines are availed, and the thicker the this line, or the heavier the line, it is at the bottom, and it tells that for digestive disease across all age group people avail. Then when it comes to skin disorder, in the age of 10 to 20 people avail Ayurveda medicine. And then endocrine and skeletal disorder, many people avail Ayurveda medicine. So this actually is telling. This is telling that if so many people are taking this medicine, there's something in the medicine. But how does it work? We do not really understand because the principles are very different. So, in today's time, what people are doing, and because everybody wants to move towards understanding health and wellness, and understanding how we can calibrate itself. Many of you might have apps which tells you drink water, take walks, do meditation. All the health apps are there. So the idea is that if you collect enough amount of information on yourself, on your data, across various sensors which you wear or various lab tests which are done, and past history, and these are all aggregated, then this becomes a big data space. And this, if you keep on measuring in spatiotemporal dimension, then a person's pattern could be understood, and through that pattern you can create a digital twin for yourself, so that twin, you can do some experiment on the twin, simulate something, and then it will tell you if you are moving to this place or moving to that place, how should you kind of reset yourself or manage yourself. And this is what is the aim of today. And these are the kind of studies which are being done in Western population, that collecting millions of such data on millions of such people, and creating using big AI based framework, understanding patterns, so one can actually create digital twin versions of themselves. But to do that, there's so many things to be done. And one of the major problems which the modern medicine on the genome, after the genome project, everybody understood, is that we still do not have a definition of how do you define a normal. For example, when the human genome was sequenced, it was thought that if we have one reference genome sequence of 3 into 10 power 9 base pairs, then at every point you know what nucleotide is there. And if you compare it with another person, let us say, who's diseased, then you might know which are the regions which have contributed to the disease to that individual. But the most surprising thing which came out after the Human Genome Project is that there are so many variations in the human genome. It leads to endless combinatorial possibilities. And so, if you look at the genome sequence itself, you will not be able to know who is a normal human. The other thing is that even there is no clear cut definition of how do you define health. For example, most of the time we define health if we don't have an illness. But if we don't have an illness, but we still want to define how close we are to certain illness or how protected we are from certain illness, we do not have that definition with us. Uh, uh, some people are getting into certain kinds of predictions. For example, there was this paper which came out in 2009 in Proceedings of National Academy of Science, which sounded much like our palmistry and all, where they said that if you take the ratio of the second digit to the fourth digit ratio, then it could actually predict a whole lot of things. And one of the things which they could predict was how likely you are to be a high frequency financial trader. That means your risk taking ability could be just predicted from your second digit to fourth digit ratio. How is that possible, right? Now, this was a immense big study which was conducted amongst financial traders in UK. And the reason behind this was that this finger length becomes longer in the prenatal stage, when a fetus, if they have a high prenatal androgen, then that androgen governs the development of this finger length, as a result of which there is a longer fourth finger. And this longer fourth finger actually predicts increased risk preference, rapid visuomotor scanning, physical reflexes, and it is basically a surrogate for circulating testosterone levels, which during the developmental time actually gave rise to a longer finger. But now this has been also linked to large number of diseases like atherosclerotic plaque and autism, attention deficit disorder, eating disorder, aggression. So the West is ready to believe this finger length, by of course scientific basis, that it can predict a whole lot of risk associated with this finger length. Now compare this to 3500 years back. There also was a explanation that body constitutions are fixed at birth, and the constitution is defined in a very kind of composite manner. I'll come to that, and that predicts an individual's predisposition to disease and all. So this is what is given in Prakriti, how it's formed during prenatal environment. And in Ayurveda, they describe diseases as vyadhi, and then they would describe diseases as whether they are curable or not curable, easy to cure, difficult to cure, palliative. If it is not curable, whether you can have palliative version, or if it is non treatable, then you kind of leave it alone. And then there are descriptions about uh, uh, kind of how you would predict there would be a good outcome in a person when the person is suffering from disease. And it talks about these four arms. It's called Mahachatushpad, where they actually describe that the physician who's treating should be knowledgeable, skillful, experienced, hygienic. Medicine should be abundant, effective, multiple preparation, compatible. Then the nurse should be, the caregiver should also be there, who should be knowledgeable, skillful, empathetic, and hygienic. And the patient should have a memory, should be obedient, should have endurance, and should be able to communicate properly, so that the history of the disease can be actually catalogued. So such kind of amazing detailed description, which the modern people are also still trying to understand, is actually vividly given. And it also talks about sperm, ovum, the what you get from mother, father. So what I'm trying to get at is that sometimes we write it off as something which might not have any documentation, but actually there are a very detailed documentation. And then it talks about uh, how do you understand the origin of the disease or the mechanism of disease. It talks about there's environment which actually vitiate something called doshas, and those give rise to the origin of the disease. And then it can lead to settlement of the disease, so that you start to see signs and symptoms. And then they have also described stages of intervention. They call it as shatkriya kal, means there are 6 stages of intervention possibilities, in which if a disease is progressing, they can identify which stage of the disease is there, and appropriate preventive mechanisms can be thought through. So, this is just like what we are envisaging as the pre womb to tomb kind of a thing, but it is all there for different kinds of diseases are described. And then, how do they describe all of this? So, this dosha is what is the fundamental organizing principle. So, just like we talk about DNA makes RNA makes protein. So, DNA is the organizing principle which will determine what RNA you will make, what protein you will make. So, in Ayurveda, an equivalent organizing principle is Tridosha. So, Tridosha is something which, if you want to understand in modern terms, it actually governs physiological homeostasis, and it has got 3 components. One of them is called Vat, one other is Pith, and third is Kaph. So, Kaph governs building, Pith governs metabolism, and Vat governs movement. So you make things, you build things, you move things, you break down things. So these are function, meaning these there are 3 kinds of broad functional entities which govern them. And uh, this organizing principle is also there, pervasive. So an individual can have different proportions of Vat, Pitta, Kapha in them. So as a result of which, some people might have more of Vat axis, some people might have more Pitta, some people might have more Kapha, and that might describe what that individual is constitution is to be. And what is very interesting is the diseases are also described as the relative movement of the dosha from their homeostatic state. So, for example, if higher state of Vat is normal for a Vat individual, movement of Pith in the Vat dimension will become a disease state for the Pith. And movement for the Vat in Pith dimension, which is otherwise healthy for a Pith individual, will become a disease for Vat. So Ayurveda looks at people's homeostatic proportion of doshas, and they then look at what is deviated in the person in the disease, and accordingly they will choose a medicine which also is described in the context of whether it will modulate Vat, it will modulate Pith, it will reduce Kapha. so that you are bringing a person to their homeostatic state and not everybody to the same homeostatic state. So this is the way, the principle in which the personalization happens, and this also forms the way by which medicine or personalized medicine is practiced. So you look at the causes, like for example hypoxia will trigger vat, cough or dehydration will trigger vat, and then you look at the features and then accordingly you have a therapy. So this kind of a network concept is in Ayurveda called as the three threads or three sutra concept. And the now this can sound abstract. What will you define as kinetic? What will you define as metabolic? What will you define as potential? So there are descriptions, and now this will sound like a standard biochemistry book, but believe me, these are the terms which are given in Ayurveda. That Vat governs shape, cell division, proliferation, signaling, movement, transport, cognition, networking, excretion, initiation, etc. Pith governs, it is the metabolic component, so it governs digestion, metabolism, thermoregulation, vision, host surveillance, execution, and Kapha governs growth, storage, strength, stability, lubrication, perseverance, etc. So these are the attributes of Vat, Pitta, and Kapha. And what is very interesting is that it also talks about how these doshas can actually govern individuality. So here it talks about an individual is not only the contribution of relative proportions or what it gets from parents, but also dependent on the exposure in the during fetal development in the mother's womb, which will govern what prakriti the person will be, and that will determine how the person is going to respond in different time, different space, different age, different season, etc. So here there is a vivid description, and my 25 years has gone in only around these shlokas. It talks about the genetic component, which is Shukra Shonit Sanyoge, Kal Garbhashay Maturahar Vihar, that's the mother's uh, diet, lifestyle, and season of conception. And then Jati Prasakta, what runs in ethnic population, Kul Prasakta, what runs in the family, Desh Anupatini, which geography you come from, time, Kalanupatini, Vaya Anupatini, and then is the individuality. So, so this actually says that if you are seeing a diseased person, first you should understand all these and contextualize them. Only then you will understand in which trajectory the disease is and what are the governing factors. So, such kind of thing from principles to human individuality of Prakriti is at this point of time not really such vividly described in the modern times. Also, uh, it actually talks about, so Vat is there in an individual of a Vat Prakriti, but these this, since this is a common organizing principle, it also has different seats. So Vat, the seat of Vat, meaning, because these are all homeostatic entities, you everybody will have to have this. So it functions more in the brain, lower gut, bone tissue, ear. Pitta works more in these tissues, Kapha more in these tissues. As a result of which, if a Vat individual will have more Vat in them, then it will transcend to their phenotypic characteristic of the way they move, the way they walk, their memory, their bowel, bladder movements, body frame, auditory memory, or if they are more pith, then they would have more overt phenotypes which will be characterized in terms of the skin, liver, blood, and eye aspects. Or if it is cuff, then it will govern build and all. So that way, uh, just like in modern sciences, we describe people on the basis of anthropometric attributes, on the basis of how they tan or in the sun when they are exposed to sun, or how their circadian rhythms define whether they are morning larks or night owls, or their physiology or their metabolism. Similarly, these Vat Pitta Kapha has got features which are composite of all of them. So if you look at Vat, Pitta, and Kapha, they will have uh, these people will have thin frame, medium frame, broad frame. Like that, there are distinguishing features of each of the Prakriti, and that's how an individual can be, in a composite manner, be assessed by Ayurveda doctors to be able to say who is Vat, who is Pitta, who is Kaph. And once you have assigned a Prakriti to a person, which can be done right after birth, it doesn't change across the lifetime of an individual. So this is like a signature of an individual, and it governs the health, the response, the disease trajectory, and how do you manage your health. So, distinct diseases are ascribed, that what people will have more of neurological, speech, developmental disorder. Pit people will have more skin, bleeding, ulcer disorder. So these are all the disease descriptions there. And cough will have more of these atherosclerotic, obesity kind of conditions. And then there is a way by which you have to evolve a medicine, and which they call this as yukti, so that how they take disease as moving targets, and as the individual progresses to disease or takes medicine, all of that has to be recalibrated, taking into account the person's prakriti. So, why I was telling so much is because there is a fundamental understanding of personalization from an individuality perspective in Ayurveda, which is the need of hour in modern medicine. So, where modern medicine has more objectivity, we know what kind of molecules are used to treat imbalances, but they primarily target the symptoms, and they treat people in an organ specific manner, whereas Ayurveda treats people through understanding doshic imbalances, and they aim at reversal in a systems perspective. But their medicines are very complex, and the goal is to have an holistic and an individualized approach. So both of them have their advantages, but both of them look in opposite direction. And what we need to now do is, if we can actually enable a crosstalk between them, so that an individual who is suffering from a disease is able to kind of take a decision where both of them crosstalk, and we get more personalized and more kind of evidence based option. So this is what integrative medicine is thought to be. But how do you make this crosstalk? Because one is written in Sanskrit 5,000 years back, and another is totally state of the art modern. And if we have to bridge these language, there are no curriculums where both of them are taught together. So how do you bridge the language? So if it's becoming a little heavier, I just say, I don't know how many of you might have read this book, or how many of you have read Jhumpa Lahiri. She writes very beautifully, very lucidly, and this is one interesting book called In Other Words. Jhumpa Lahiri is an Indian. She settled in Canada, and she writes very vividly. So as you're reading, you can visualize what she's saying. Now, in the middle of her career, she decided to relocate herself to Italy, learn Italian language, and start to write books in Italian. So this book, in other words, is her biography of mastering Italian language. And if you read this book, you can understand what pain she would have gone through to master a language when your native language is not that. But what is very interesting after you read the book is you understand that she has actually written this autobiography in Italian after mastering Italian, and another person has kind of translated it, and when you read it you feel that it is Jhumpa Lahiri's writing. So this could only happen if she's got under the skin of Italian language, so that she's been able to articulate and translate in such a manner that anybody who reads the Italian version is able to translate it in a way that the English version looks like Jhumpa Lahiri. Why I'm trying to get to this is because this is what we want to do, that we do not want to just do just a translation of a Sanskrit to English when we want to take Ayurveda to mainstream. We want to understand Ayurveda in its from the foundational principle, so that we can iteratively build the knowledge and we can make it more operable. So this is what we thought 25 years back, that could, is there a possibility that what these pith, kapha and vat is there in Ayurveda, could we actually have a molecular description of this, because molecular description is what will then connect to modern description. So for example, if individuals are more prone to hemorrhagic disorder, kapha to obesity, vat to neurological, so if we take individuals of these prakriti type and look at their genetic makeup, will we find more kind of signatures for these disease in the different individuals or different groups of individuals, or would they have differences with respect to transport function, storage function, metabolism function? So that was our hypothesis. So we thought that, can we take people who are at the end of the spectrum? There are normally seven kinds of people, some who are extremes, and most of the people would be middle, vat, pit, kaf, like that. But if you are most extreme, then you are more vulnerable to certain kinds of diseases, and they would have more extreme phenotypes. So we thought we'll start with extreme phenotypes, take the blood sample, and try to study their blood sample expression or biochemistry, and see whether they differ if we take these groups of people. So this was our first study. So what we did was, we took individuals, healthy individuals, and now we had to do many things, that we should ensure that these are not coming from different parts of the country. So whatever differences we are seeing is just because of the difference in country and nothing to do with prakriti. So they had to be same age group, they have to be from same region, etc. So once we were able to do that, and lots of things went into it, I'll not get into the detail. What we did was, we classified them, identified predominant prakriti, and did their routine biochemistry, whatever we do from, let us say, Lal Path labs or any other labs. And then we took their peripheral blood and did this whole genome RNA profiling, which is called transcriptome profiling. And then we also looked at genetic differences in a set of genes. And then we try to see if they differ, and if they differ, what do they differ in, and can we synthesize this information, and then try to see how much does it resonate with Ayurveda. So this was the work which we initiated. And so there was a genetic map. I was involved in making this map with a consortia of people. It was called as the Indian Genome Variation Project, which told us that people, if you collect samples from this part of the country, then they are more similar to each other. So this is a genetically homogeneous group. From here we identified individuals from 18 to 40 age group, classified them using a Prakriti questionnaire, and then took individuals who are at the end of the spectra, identified the predominant Prakriti, and then did their analysis. So what did we see? So this was published in 2008. The first striking thing which we saw is that, normally when we do any biochemistry, we have a range, and if we are within the range, we all feel that we are healthy. But if we stratify them by Vat, Pitta, and Kapha, then there are ranges within the range which is allowable for certain Prakriti type. So for certain people, certain parameters, Kapha individuals tend to be in this range, and maybe other would be in the other range. So this Kapha movement within this range to a little higher level might be disease state, but would not be so for Pith, and might not be so for Vat. And then this would need some movement here by a little increase in Vat, but Kapha will take a longer time. So that shows that even if we use routine diagnostics, there are range within the range which is allowed for individuals. So like that, we studied what are those differences, and we found things like hemoglobin, RBC, prothrombin time, those differentiated with from Vat and Kapha. Lipid profiles were higher in Kapha, and then serum prolactin, HDL were higher in Vat. And then when we do the expression and all those kinds of analysis, we find immune response to be higher in Pith, cell cycle to be higher in Vat, which was very interesting. Ayurveda has so this was what we published, whole genome expression and biochemical correlates of extreme constitution. But this is also what we published, that Vata regulates cell division and morphogenesis is actually given in Sushruta, and there is a vivid shlok which talks about structure, components of body at the micro level, parmanu bhed, aparisankhya, which are the innumerable, ati bahut, very subtle, you cannot see by naked eye, and they can join and they can proliferate, and the whole function is because of Vayu, or which is Vat, and that is its functional attribute. So, this was really amazing that what we are seeing now actually has also got a correlate many years back, right? Now, then we thought that if it talks about this, and here we had taken peripheral blood from individuals of Vat, Pitta, and Kapha, what happens if we take cells from individuals and grow them in petri dishes? Is their behavior recapitulated? So, what we did 10 years later, uh, we grew the cells, because there are these stem cell technologies where you can take a peripheral blood, immortalize them, grow them in petri dishes, and then count how the cells proliferate, etc. So one such experiment we did on biological replicates. That means we took three individual vat of three individual pit, three individual cuff, and tried to see their doubling time, that is proliferation. And we see that Vata and Pitta double faster, at a faster rate compared to Kapha, which takes longer time to divide. So this was very interesting. But then we asked a question, why does Vata divide faster? And Vata also has a very lean body frame. We would tend to think that a person who is a more broad frame like Kapha would have more cells in the body, so they would have more inherent cell proliferation rates. But it was counterintuitive. We found the observation was counter to that, and we found more proliferation in those people who have a relatively thinner body frame. So then what did we do? We actually gave them UV, that's a damaging agent where there's a lot of death, and we found that there was more number of dead cells in Vat compared to Kapha. But if we waited for the cells to recover, then the numbers became the same. So which told that maybe Vat are more prone to DNA damaging agent. So inherently they have got a machinery which allows them to replicate faster. So it's a trade-off, but this trade-off can have a so this is what we published in a cell cycle paper, where we talk for the first time that even at the cellular level, if we classify people by prakriti, we see the differences in their cellular behavior. So this has a relevance. For example, in cancer, you give these chemotherapy which cause the death of the cells, so you calibrate the drug according to how prone you are, right? But supposing the individual is vat kind and you give it a lower dose of drug because you see more death, then in that case, if they have a more cell proliferation advantage, there might be more chances of uh, 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 kind of reoccurrence of tumors, right? So there's a speculative framework that supposing you were to classify people on the basis of prakriti, could that influence radiation sensitivity and repair capacity, and so guide the therapy accordingly. Such kind of cell proliferation defects are now being seen in many diseases. So, autism spectrum disorder, from cell proliferation to clinical phenotype, people are talking about there are special autistic children who have a bigger head size, and that head size is thought to be that during early growth, if the cell proliferation rates are higher, then they have more number of cells which go on to make the size of the brain, and so you have 67% more prefrontal neurons in autistic children. And the experiment had been done that they had taken cells from autistic children who have bigger head size compared to normal. They have similar kind of phenotype as VAD, that is, they proliferate much faster than the control. So what we are seeing by a phenotype described from ancient types, and the molecular link to those phenotypes actually resonates with what is being seen in today's kind of diseases. So it actually gives a kind of a thought that this is likely to be useful in today's condition. And then now, if you there are databases where you can do such kind of searches. For example, abnormality of cell cycle. If you ask in a particular database, it will give you certain kinds of disease which are linked to abnormality of cell cycle, like let us say Fanconi anemia and all. So these are defects of the cell cycle. And if you look at the genes, these genes will actually show that these are involved in repair, DNA damage, DNA cross-linking, something which kind of resonates with the function of Vata. So here is a way now we can start to think how we can connect the languages from one to the other, so that maybe we will be able to port certain kinds of information. So there are other kinds of discoveries we made. One very striking discovery we made was that if we classify people by prakriti, we actually find genes which differ between them, and such genes become very, very relevant in certain kinds of condition. So for example, we discovered an oxygen sensor gene. So when you are in low oxygen condition, your body has to recalibrate into for coping with low oxygen. Like when you go to high altitude and oxygen levels are low, then the body needs to ramp up more of oxygen. So you have angiogenesis, more hemoglobin made. So those are all the conditions, and it will depend on how quickly you are able to switch to that. You will be more be able to cope up with such low oxygen condition. So what we found was that this gene differed in expression and at the genetic level between two prakriti type, such that we could predict that cough is cannot cope up in hypoxia, and pith can cope up much better in hypoxia. Now we found that uh, natives who reside at very high altitude, that is Leh Ladakh, had the same genetic makeup as we saw for pith. So then what we thought was, could then we use the genetic marker which is associated with cough to predict that who are the people who go to high altitude and develop certain kinds of heart conditions, they would be more like cough type. So this is what we tested. We tested in a set of level dwellers who developed high altitude pulmonary edema, the cough genotype, and we found them to be overabundant. So this was the first time we could actually show that if at the sea level you classify people by prakriti, you might be able to predict who are the people who would do better at high altitude and who are the people who might have more problems in high altitude. And Ayurveda also describes high altitude. It assigns prakriti to high altitude, and it also talks about if you go to very high altitude, then there are possibility of kaph vat kind of a problem in high altitude. So there is a kind of a recommendation of a particular kind of a prakriti who might be much better habituated to high altitude. So this was one of the prominent studies which we could show that if we classify people by Ayurveda, we can actually identify or discover genes which are linked to high altitude adaptation. And this discovery has been validated in multiple altitudes of the world: Tibet, Tajikistan, Andes, Africa. And this is all around the time in which we published our paper in PNAS. And recently, people have shown that this is also linked with aerobic capacity. So, as a result of which, you can predict endurance athletes and sprinters who have different oxygen requirements could also have such a kind of a difference. So, could Prakriti govern differences in our ability to have get into different kinds of sports which are linked to aerobic capacity? So, over a period of many years, we've been able to show that if we classify people by Prakriti, then we can predict the oxygen coping mechanism. And this was taken up by a clinical collaborator, Dr. Anurag Agarwal. He said that if oxygen is involved in high altitude, it could also be important for conditions like asthma, where lung function is compromised. So, supposing you have this kind of a pith like state in asthma or cough like state in asthma, what will happen? And it turned out that when they did this experiment in mouse system, where they made by small molecule they kind of mimic the condition of hypoxia, that is pith kind of condition, then it brought about more inflammation and epithelial injury. So the mouse's lung function was compromised. They became steroid resistant, and then their mitochondrial shape function all got compromised. So this showed that if asthmatic people have differences in hypoxia responsiveness, then there is a possibility that they might actually have different kinds of inflammation outcome. Now, this became a very good test bed for testing Ayurveda medicines because, as I told you, Hetu Ling Aushad. So, we can say if pith cough is perturbed and if there are medicines in asthma used for such perturbation, if we give it to such mice, will it actually, first of all, reverse asthma, and will it reverse by modulation of hypoxia? So we can test it. So this was a study which was done by one of our students. There was a medicine in Ayurveda called Adatoda Vasika. If people take Ayurveda medicine, you'll see something called Vasa. So this medicine, when we gave to mice, all the things reversed back. Inflammation, steroid resistance, mitochondrial shape, mitochondrial function. So which actually closed the loop that maybe once you understand this, then you are able to kind of rationally use Ayurveda medicine if such a kind of a loop is there. And if that were to happen, then whichever other places where hypoxia is compromised, could you also start to use Adathoda Vasika, which is what was done by the student further. And they could show in preclinical mouse models of COVID, Adathoda Vasika could attenuate inflammation and hypoxia, which now tells that if you have a newer disease today, how would an Ayurveda doctor kind of choose the therapy so that it actually attenuates the symptoms? So this is the whole framework of Trisutra, but now as you can understand, it's in a molecular language and it is iteratively built. We find hypoxia, we test it in modern model, and when the model gets recapitulated, we can use Ayurveda. So you can meaning iteratively do this, and this is what we call as Ayurgenomics. That genomics has provided a molecular language to Ayurveda so that now you can start to have conversations between them. So this is the framework, operational framework, that you have principles, you develop a working hypothesis, test it in Prakriti or cell model, and try to see whether you can get the correlates with dosha. And if you get that, then can you test it at genetic level? And if you have, then you can correlate association of phenotype, susceptibility, and responses to doshas. And if that was so, then you can perturb that in model systems like mouse. So you have a model of mouse of Vikriti, which is the from Prakriti to Vikriti. And then if that is there, then you can choose the drugs which can be tested in mouse model. And if that closes the loop, then you know Tridosha is an organizing principle, and that can form the basis of repurposing. So this is the entire framework, like I showed you. And like this, many evidences have been accumulated over the years, not only in transcriptome, but metagenome, gut, proteome, biochemistry, and by other groups also. They started, and this Ayurgenomics has become a subject in the country. Now, the other things we have done, that supposing we find that this Trisutra works in an iterative manner, then we can take a drug of Ayurveda and use it to probe linkages in causes and feature, as a result of which, if we want to find new drugs for new diseases, then using this same framework, can we actually discover or repurpose medicines. So, such a kind of a framework we have developed, and we demonstrated it in a medicine called Cisamplus parera. It's a medicine which is used in Ayurveda traditional medicine for snake bite, hormonal disorder, sepsis, bleeding, tumor, digestion, dyspepsia, malaria, etc. And it is present all over the world, more in malaria endemic region. And we wanted to study whether this drug, if it works against so many things, is it that because it has so many molecules because of which it works, or there is a common underlying kind of a mechanism which links to all these diseases, so one medicine can then be repurposed for another condition. So we tested it out, I will not get into the detail, but just trying to give you the framework that the same thing we probed through genomics, whether disease A, disease B, is there a molecular connectivity? And we ended up finding that there is a connection through the estrogenic axis and dengue inhibition. So that's why it can be used in diseases like breast cancer and also inhibition. So there was this thing. But not only that, because it inhibits dengue and then COVID had come, so we tested whether it could also have possibility for usage in COVID. And we actually did studies and we could show that uh, by studying it in models of SARS-CoV-2, that is infection models of SARS-CoV-2, giving them Sesam plus Pereira, we could actually show that it can inhibit COVID titer by 99%. So that tells that not only whatever it talks about you can discover, but you can actually use it for emerging diseases like COVID. So it actually tells that Ayurveda is contemporary, and if you have a Ayurgenomics kind of a framework, everybody knew how we all kept on measuring our oxygen, hemostatic outcome, and these are all we observed earlier in 2010. And then a clinical trial was done in COVID patient by Dr. Bhavna Prashar, with whom I had collaborated for 20 years. She's an Ayurveda doctor, and she did a clinical trial with All India Institute of Ayurveda and could show that this drug was also useful in mild COVID patients, in especially in the hypoxia. So this is what it shows. So Ayurgenomics thus can be used in identifying molecular correlates of dosha, why people are different, why they differ at the genetic level, why they respond differently to drug, how you can stratify patients, how you can get actionable targets, how you can identify new molecules for repurposing, use them in predicting why people differ in their health outcomes. And now, once this foundational thing is ready, can we start to use these foundational principles into in an integrative medicine setting where the two doctors can cross talk by such kind of an evidence. So I'll just briefly tell that having these kinds of things, we decided, or I decided, to move to IIT Jodhpur because now we think that it has come into such a kind of a space where we need other kinds of people who work in artificial intelligence, modern medicine, genomics, engineering sciences, so that we can actually establish an AI driven framework for integrative medicine, because we will need to have huge data collection, build models, have an understanding, and then only we can build models which can be useful in an integrative medicine setting. So this requires a huge different attributes of AI. So this is what we have in IIT Jodhpur. And here what we are doing is, we know the foundational principles are sound, but now we are linking it with data science, engineering science, multi-omics, to be able to see how we can translate it in integrative medicine setting. For example, one such example. Do I have some time, Shilpa?
Yes,
Hello?
ma'am. Yes, ma'am. Please continue.
Okay. So this is one such thing. We I will just give you a flavor. There are other things which are being done by other investigators. This is in the area of rare diseases. So most of you would know rare diseases are very severely debilitating, but because they are rare, people take a lot of time to diagnose it. There are limited treatments available, and whatever treatments are available costs crores and crores of rupees for managing. People are not interested in research because it's not very incentivizing, neither for the drug company nor for the researchers. And then lack of awareness, right? And this is because there are 12,000 rare diseases. They get distinguished by the system, like your skin diseases, eye diseases, renal diseases. They all go to different specialities and they are defined by organs. Now, Ayurveda has a very interesting framework. It's called Anukta framework. Anukta means that which is unseen, unheard. So 5,000 years back, they documented this framework from in the context of saying that there could be many, many more diseases which they have not encountered 5,000 years back. For example, COVID, or many medicinal plants which they are not seeing now. Let us say avocado, right? But if they are present, and if Ayurveda were to understand that, how will they understand it? What they have not seen. So they actually have a framework as to the things which are not seen, but which we might see in a future scenario. We can understand it from its foundational principle, and then connect through the foundational principle to the foundational knowledge, and use a reasoning which they call as yukti to be able to evolve a medication which will be contemporary. So such a kind of a vivid description is also given for what is not seen. So we decided to ask a question, that could the rare diseases be treated through such a kind of a framework. It's a new thing. A disease like Bardet-Biedl syndrome, myotonic dystrophy will not be explained as such in Ayurveda. But if we break down this disease for if from an Ayurveda perspective, can we actually understand this disease from Ayurveda and try to see whether Ayurveda can provide a kind of a treatment modality? Can we explore that? So this is what we did. So, as I had explained, Ayurveda describes diseases in terms of vikar. They call it as if single dosha is involved, then it's called nanatmaj vikar. If multiple doshas are involved, then it's called samanyaj vikar. And there are 80 kind of nanatmaj vikar of vat described. Uh, 40 kinds of pith nanatmaj vikar are described, and 20 kinds of kaph nanatmaj vikar are described. So now how do you connect it to modern descriptions? So there is a interface available. It's called human phenotype ontology. So supposing there is a abnormal bleeding, if I give a query, then it lists down syndromes which have this abnormal bleeding as the symptom. So these are syndromes. Now, each of these syndromes will have many features in them: body, size, shape, many, let us say 17 to 30 features. Now, if we give these features to Ayurveda doctors and ask them to label it as whether these features are Vat, Pitta, and Kapha, and we do it systematically with all the diseases, then we get proportions of doshas in each of these diseases. Now, what you can see is red coming out very overtly in the proportion in all these diseases. So each of these diseases classified by Vat, Pitta, and Kapha. And now, if you look at this disease from the lens of Ayurveda, they will convert it into a shloka called Rakt Pit, because all of them primarily has a pith involvement, which is the red color. So we thought that if this is possible, then could we do it for all the diseases? So there are 12,000 rare diseases and 10,000 such features. So we sat with Ayurveda doctors to start to connect each of these features to doshas, and then do a clustering, a mathematical clustering. So this whole process is ontological engineering, where we are bridging the foundational roots through unifying terminologies, right? So when we do this such kind of a clustering for 12,000 diseases, we end up finding groups of diseases which are kind of aggregated by Ayurveda. So, for example, 489 diseases which are otherwise very segregated organ wise actually come under one umbrella if we classify them by dosha, and all of them are mostly pith kind. Or if you look at another one, then it is mostly vat kind, or something is cough kind, ciliary dysfunction. And there are these diseases which has genes, and if you look at the genes, then they talk about that this cluster has diseases where DNA damage and response is primary culprit, metabolism is primary culprit, immune aging is culprit, ciliary function is culprit. So now what you see, so many rare diseases seen as rare becomes common if looked at from the dosha perspective. And now if you think of that this has more inflammation and pith kind, then if you have medicine, you have other kinds of modalities to reduce pith, then maybe you can manage this disease in a better manner, because once you have these disease, it is a lifetime problem. So this is what we discovered, that we can cluster the diseases based on Ayurveda phenotype. And then can we do Anukt Vyadhi interpretation? So supposing this is one disease which is given in modern terminology, if we break it down to Vat, Pitta and Kapha, we find that predominantly it is Vat, few Pitta and Kapha. And from Ayurveda perspective, that is the Anukta framework. It talks about, look at the nature of disease, look at the place which it manifests, look at its etiology, and you will be able to understand that it is primarily a Vata disorder, because it is involved in so many organs, and it has mutations, and it is genetic in nature, and it can only be managed but not cured. So what will an Ayurveda doctor say? That this disease is primarily of Vat kind, Pitta and Kapha are later manifestation. But for them, Vat increases with aging. So many of these are age related also. So if you halt the aging component by particular therapies, Rasayan therapy, Panchkarma therapy, could you actually manage this disease so that a person has having a better quality of life when he is living with such diseases? So this is how we now visualize that if we do it in large scale, then we might be able to have a future for patients who have chronicity and who can be managed. So this is my last slide, that hopefully I have been able to show you how we can have a next generation health solution by integrating the biological intelligence understanding by Prakriti and Vikriti through a natural intelligence of dosha and trisutra. That is, in any chat GPT you do, it has to learn from patterns through using natural intelligence. And if you are able to weave these, then you can use the Anukta framework, which is not what artificial intelligence does, to be able to get a proper inference. So Ayurveda knowledge system, it is inherently generative from foundational principles of dosha and trisutra. And Prakriti and Vikriti could be data models which builds for to build the system for understanding spatiotemporal dynamics. And creative inference can be made using Anukta framework for newer scenarios, and Yukti can provide an algorithm for synthesis of knowledge. So this could actually be just like new brain has kind of inspired neural network. So could you actually weave the fragmented understanding of knowledge through the foundational framework of Ayurveda to be able to have a much better understanding of health and disease and managing health? So I would like to now kind of conclude here. And this is the team in IUTech. We are doing many more things, and multidisciplinary electrical, computer science, chemistry, and working with Ayurveda doctors across the country to build this framework. And this has its foundation in IGIB. Professor Brahmachari, he visualized this kind of an area. He's been our mentor. Bhavna, with whom I worked to develop this foundational framework, and large number of students and collaborators from different areas, modern medicine, Ayurveda doctors, computational biologists, everybody put in their part to be able to build this area. So thank you, and I welcome you to this beautiful campus and open for questions. So
Thank you, ma'am, for such an enlightening and enriching session. Uh, Participant, you can raise your question in the chat box.
I don't know whether it was okay for everybody or I went very fast.
No, no, ma'am. Actually, I wanted to compliment you, ma'am. Especially like how, like personally, I just want to know this Ayurvedic language is so complex. Like how your journey started. Like how could you correlate? Like for that, you need to be well versed with the Ayurvedic text. Then only you
So
can think of such
So I
experiments.
have not read Ayurveda text because I am not competent. I have not been trained. I did my PhD in hardcore molecular genetics,
Mhm.
and only after I joined IGIB that I got to hear about uh, Prakriti Ayurveda, and I didn't know where to go because my boss came from a meeting and he showed me this triangle, and then luckily I had a neighbor who was from ancient history, and I told him I've been asked to find out about this, and then he ancient history means he knows Sanskrit, so we gave him some fellowship kind to be able to give a English translation, and from there we understood, oh, this has got so much knowledge. Then we started looking for Ayurveda doctor and met Dr. Bhavna, and we started building this conceptual framework and then went on to this journey. It has not been so easy because there are so many biases in such area. So so this is where how we started very long back. So it had its own ups and downs because to be talking about this in a modern setting, which was my peer group, was a challenge till we had, after 7 8 years, a PNA's paper, which
Mhm.
was kind of acknowledged that if Western people are acknowledging this, then our science is okay. That's how we typically have grown in our country. So, Western credibility was important for us to understand that there is this is scientific, and that's how we moved, right? But
Yes, ma'am.
it's been organic growth and lots of people joined in the journey. But it could have been much, much more. 25 years is a long time, but still, now lots of people have come on board and they are wanting to collaborate. And this is becoming a subject in itself.
Yes, ma'am. Ma'am, also I wanted to know, like, there one in one slide you showed that different prakritis, like for example, hemoglobin. You said that for some prakriti it was different, but when
Hematocrit is higher. Huh.
sorry, so for normal healthy person also, even if the prakriti is different, the hemoglobin will vary. This is what I understood. Am I correct?
If they belong to different Prakriti group, their hemoglobin levels might differ. What is the normal range?
Yeah. So, ma'am, when we get our blood tested, like Lal Path, they give us some range. Like, for
Yes.
example, 12 to 15 is considered good. So, like, when you tested on these individuals, you got these levels within the range?
Within the range, they are a range within the range. So if you
Okay.
test yourself for many years, you will find that you always tend to remain in one side of the range. You are not
Okay.
all over the place, right? That
Mhm.
means that is your range. And if you go a little over it, then that is an alert that something is moving from your normal homeostatic state.
But in modern health, the entire range is healthy.
Range is a range. So there is no breakup of, because there is no definition of health. All times we describe health is not within the range, then it is, meaning not outside the range, then it is healthy.
Mhm.
That's what how we describe in modern, right? But in
So,
Ayurveda, it is everybody is in a different state of health, and they could be more prone towards something but more healthier towards another thing. So you need to know in which place you are to be able to say when you go to a disease state from where you have moved.
So, ma'am, again, coming to the genetic level, we all
Yeah.
humans have same DNA in the sense that enzymes will be same in every person.
But all the enzyme activity will not be the same because we have genetic variations which might
Yeah.
make our enzymes differently active.
Yes, ma'am. So, for example, if okay, the maybe the expression is not that much. This is
Yes,
what you want to say.
yes.
So, one question is in chat box. Can you tell some tools of AI to create a knowledge graph? Are the tools freely available?
So people are using NLP, LLM. I collaborate with AI people, and they use the tools, and many of the tools are freely available, as I understand. I will not be able to tell, but if the person is interested and writes a mail to me, I'll connect it to the knowledgeable person who does AI and NLP.
And one question is written, but I'm not able to understand. Is it possible dosha shifts? Maybe the sir can. Okay, you can.
Yeah. So Prakriti doesn't change, but doshas. So, for example, you somebody asks you blood pressure, right? And you give a particular value, but if you measure yourself all through the day, you will have different values of your own blood pressure. Then how do you define what is your right blood pressure? So that is something which you measure at one time of the day and every day. Then you have a steady thing, and during different times of the day your pressure fluctuates because of diurnal rhythms, right? So since doshas are functioning entity, they also have temporality. So for example, 2 to 6 in the morning and 2 to 6 in the night, vat increases, and after 6 to 10, kaph increases, and 10 to 2, pith increases. So Ayurveda doctors, if you go to them and say you are having fever, they'll ask you what time of the day you are having most fever, and then from there they will diagnose what vikriti you have, whether it is vat, pith, or kaph. So that is vikriti. And then there's something vaikarik, which is temporal every day, but it is just like a routine shift. And then there's prakriti, which is your baseline state, right? So your baseline state, if you are born and then you age, it will not change, but definitely you change, you age, right? So everybody ages, so that aging component is contributed by vat. But if it is in the vat individual, he will prematurely age compared to the other prakriti. So dosha proportions will change, but they have to be contextualized by your baseline dosha, so your prakriti doesn't change.
Okay. One more question is, can you give some takeaway for common layman from the Ayurvedic treatment so that we could find dosha and apply in daily routines?
So now that you know that there is this dosha thing, uh, only my only thing is, if I've given you the survey, and first of all, how many of you think that having turmeric as a spice and having turmeric as a medicine, both of them are the same thing as an Ayurveda medicine? How many of you feel so? If I go back to the survey, it will be like so the next question will be, do you think turmeric could have a side effect? Right? So most of us would think that turmeric is in all our food we use turmeric. So how could turmeric have a side effect? And since turmeric come from Ayurveda, then you will say if you started eating turmeric, then you are into Ayurveda, right? Both the things are wrong. Turmeric can have side effects because turmeric works on a particular dosha, and if your doshas are elevated to different extent, then you need to take care that you are not overdosing yourself. And similarly, if some neighbor of yours comes and you have taken a medicine from an Ayurveda doctor and it helped you, and your neighbor comes and tells you that they've got this problem, then if you've gone to a modern doctor, you never share your medicine with the neighbor, because you will tell that this is the doctor I went to, you go to this doctor. But if you have got an Ayurveda medicine, you will hand it over very easily to the neighbor. So you, because you think that this is not medicine, it is churan chutney and it can be shared just like that. So that's where the problem comes in, that Ayurveda is also a system of medicine. Of course, there's some over the counter thing you can have. But if you ask an Ayurveda doctor, should you have methi every day, like everybody is consuming methi who have diabetes, then it will tell if you are a pith prakriti, you should avoid methi because it has a heating component which will elevate your pith. So these are the kind of things you need to be aware of, and these are the things which actually having an understanding of a dosha might enable you. So I hope if you go and read any Ayurveda book, you will read it more cautiously and much with a much clearer understanding of Vat, Pitta, and Kapha.
Any other question we have? The participants are requested to ask, or you can type in the chat box. I think there are no other questions. Uh,
Okay.
But ma'am, I would really like to thank you for enlightening us on this topic. I personally use Ayurvedic products, but now today's talk has further, I have started appreciating it more. The Ayurvedic. I'm glad that, because the only loophole, like we as Indians don't give value to Ayurveda. But if we have more publications and more scientists like you who do these studies, certainly this will gain momentum. Ayurveda.
Yeah, we would like more people to join in. And if you have biases, you think it doesn't work, then test the biases with a method of science and then try to write it off, is what my request would be to all. Because we tend to judge the science by peer, and if we think that our peers are not saying good things about it, then there's something bad about it. If you are scientific and you have scientific mindset, then test it. You have a null hypothesis, Ayurveda doesn't work, let us test it out. But it has to be tested with a scientific rigor, and then only people should talk about whether it is not science. And I would like to make one thing very clear: with my Ayurveda, whatever I presented, I'm not endorsing the practice of all practitioners. I'm endorsing the principles of Ayurveda. So if Ayurveda doctors are following the principles of Ayurveda in their practice, then it might work. But they also need to understand today's disease by language, so that they are able to best connect it with their Ayurveda Shastras to evolve the best medication. Because one is written in Sanskrit and one is written in modern language, and an Ayurveda doctor has a double job to understand modern medicine and Ayurveda medicine to be a good Ayurveda practitioner. So that is something which you people will have to keep in mind, that just because I'm saying the science is right, you should not go and pick up any doctor or pick up any medicine and they say that it is scientific, so I should consume it. It is it has to be done as you treat modern medicine. You should treat it with that caution, and it has got lots of holistic benefits.
Ma'am, just last thing, like can you just tell us some research avenues in this area, like what all we can do
So
scientifically? Yeah.
there is a whole lot. Like I told you, all my students are modern researchers. So you can identify more molecules for drug repurposing. Today's date, 75% of the drugs are inspired from usage in Ayurveda, but nobody gives a due cognizance that it is based on Ayurveda, right? Levodopa for Parkinson's come from mucuna, or whatever they call it, ashwagandha, sarpagandha, all of these. It has given artemisinin. So many things have come. But if you use it from the rationale of their usage in Ayurveda, you will make better discoveries. It will be useful for stratification of patients from vis-a-vis health outcomes. Some of the AIMS Jodhpur doctors have started. One of the doctor has actually started looking at chronicity of otolaryngological disease like nose problems, ear problems, vis-a-vis what is their prakriti type, and they find very interesting correlation. And so Ayurveda is not using Ayurveda drugs only. There are lots of things of lifestyle changes on prakriti, based on prakriti, food based on prakriti. So you needn't say that you have to take Ayurveda drug. You follow the principle, then you will automatically find that there are more avenues opening up, where how you should do exercises. If you are pith prakriti, your body tends to heat up more, then you should do those exercises which keep you cool, because inflammation can also cause problem. So it become makes you more mindful, and that is what is talked about as participatory medicine. So we have P4 medicine: predictive, preventive, personalized, and participatory. So participatory is that you understand yourself much better, and you try to then use such things. So Ayurveda now people are using for sports medicine, many things. There are lots of avenues where it can be integrated. And AI, digital health, Ayurveda has started an AI mission in Ayurveda, because how do you understand the Sanskrit, NLP, LLM language models need to be made from Ayurveda perspective. So different avenues have opened up. That's why in IIT Jodhpur, different departments of electrical engineering, sensors, new equipment which can give objectivity to their practices. So electrical engineering, chemistry people, bioscience and bioengineering are using nanosciences and different kinds of organoid models for testing the efficacy of Ayurveda medicine in unmet meaning needs, the challenges like um, antimicrobial resistance, TB, silicosis. So there are lots of places where Ayurveda drugs are now being tried because modern drugs are not there. So those opportunities are also there.
Yes. All right, ma'am. Thank you so much for giving us your time and um, very basic conceptually and very comprehensively you explained the concept and linked the both
Thank,
the Ayurveda and modern science. Thank
thank
you
you.
so much, ma'am.
Thank you so
Thank
much.
you.
Okay.
Thank you. So tomorrow is the last day of IFFTP. So this is for my participants. The first session will start at 2 p.m. and tomorrow you have test also. It is mandatory to give test, otherwise certificate will not be generated. So I'm just posting the attendance link in the chat box. And please attend the last 3 sessions and give the test, otherwise the certificate will not be generated.